Okay. So good morning. Sorry, good evening, everyone. So I am Shobik, and I'll be taking this TA session. So I am an integrated PhD student in Indian Society, Bangalore. So this is the fourth TA session, and based on our week 5 lecture about proteins. Okay. Um. So before going forward, I would like to ask if anyone have any doubt regarding this session, and you can post. You can either turn on your mic and say, or you can post in the chat. So, should I take that there was? I'm taking that there was no doubt regarding any of the courses, like any of the topics. So then we'll move forward. So yeah. Again, so whatever I have taken in this, also this in this chapter, I have taken everything mainly from Leninger this time. I have not used Harper this time. You can also follow Harper also if you want to join. And but Leninger is the best book if you really want to learn the subject. So yeah, and okay, so okay, and so first question. So first, I'll just briefly discuss about amino acids. So sorry, I forgot to give the title amino acids. So so amino acids are basically like building blocks of proteins, and amino acids are basically building blocks of proteins. So amino acids, as you know, it has a general structure of like this. It will have a NH3 group here. It will have a carbonyl group here, COO, and sorry, carboxylic group here, and here one hydrogen, and there will be R group. Now the R group varies. I'm having a chip issue, so I'll stop this and share the screen. Here for me to do. Okay, so we'll have we have this carbonyl group here, and based on this sorry this R group here. So based on this R group, we'll uh, separate the different types of amino acids. Now, amino acids, as you can see, this alpha carbon or C alpha, it has four different like R groups or four different bond, groups attached to it. One is like the CO minus, one is the amino group, the hydrogen as well as R group. Now, if you think as same as our carbohydrates, so this alpha carbon is actually the is a polar carbon, right? So not polar, sorry, it's a is isomeric carbon. So that means it will have some till the amino acid polarity will change if you give the plain polarized light to either L amino acid or D amino acid. So since these are only two types of each of the amino acids based on this alpha carbon only can have two CU isomers. So that's why they are also known as we previously told as enantiomers, correct? Enantiomers, okay, known as enantiomers, and say alanine and D-alanine, okay, and based on the R groups, now there are several 20 natural amino acids, and based on the R groups, we divide the amino acids. So there are a few amino acids which will have non-polar aliphatic R groups, and those amino acids will be preferred in the hydrophobic cleft of the protein, when the protein will be from, like from glycine, proline, valine, and methionine. As you can see, these are all the R groups. They are all don't have any charge. Then these have some of them have aromatic R groups like phenylalanine, tyrosine, and tryptophan. Okay. Then we have polar but uncharged R groups. These are polar, like it either contains OH or SH, something like that. But they are not polar amino acids or CONH2 bondings like that. So these are polar but uncharged R groups. But again, these are polar amino acids, so though they won't prefer the hydrophobic part of the amino acid. But in certain conditions, these can become uh, charged, right? This amino acid. So then we have positively charged R groups like lysine, arginine, and histidine, and then we have negatively charged R groups with aspartate and glutamate. Okay, so these are 20 different amino acids that are there in like nature, and when a peptide bond is formed, that time the OH from the one amino acid and the hydrogen from the OH from the carboxylic group and H from the uh, amino group is taken out as condensation and forms water, and this particular bond, the CONH bond, or this bond, is known as the peptide bond, correct? So this peptide bonds are formed during formation of the amino acid, correct? So yeah, so with this as the basic ideas of amino acid, now if you consider thus only this structure with several more amino acids, that will be considered as the primary structure. Then we have secondary structure like alpha helix, beta sheets, and we also have the tertiary structures like then the 3-protein 3D structure, and we have the quaternary structure, which proteins do have the multiple subunits. So for the alpha helix, will have some helix-like structure, and for beta sheets, we'll have either parallel beta sheets or anti-parallel beta sheets, correct? So yeah, so these are the kind of like the structures, and as you know, if you go for the alpha helix of a protein alpha helix, the C1 of the peptide bond forms hydrogen bonding with the C4 here. So this is from the hydrogen bonding, and beta sheets and alpha helix, those are all formed by hydrogen bonding only, right, between the amino acid, even for the alpha helix or even for the beta sheets and uh, alpha helix. Okay. Uh, yeah. So then we come for the questions based on this amino acid. So what amino acid is like the beta carbon itself? So this is the alpha carbon. The alpha carbon, this carbon is known as the, so second carbon from here is known as the beta carbon. So this is the zero, this alpha, this is beta. Then you can go like several part, like this will have alpha, beta, this will alpha, beta, gamma, and several other. Okay. So which carbon, in this case, this will be the beta carbon. In this case, this will be the beta carbon like that. So all of the amino acids, as you can see, have this beta carbon like this. Okay. So which of the amino acids lacks the beta carbon? So someone please tell me the answer in the chat or in the uh, chat or in the whatever by unmuting yourself. Okay. So no one. Okay. Okay. So the correct answer is glycine. As you can see, as a R group, glycine has only hydrogen. It doesn't have any beta carbon, right? So this has only hydrogen. So no beta carbon. So the correct answer is glycine. Okay. The amino acid with thioether linkage. Now, what is a ether linkage? The ether linkage is basically this. So we know as the ether is R, it is R1. So if this is R1, then O R2. This is the ether linkage. Now, what is thioether linkage? In case of ether, we'll have oxygen here. In case of thioether, we'll instead of this oxygen, we'll have a S, correct? And so yeah, so thing this is the thioether linkage. We have a S, and one side one R group, other side other R group. So now which amino acids contains only like which two amino acids only have sulfur in their side chain? Okay, so there are two amino acids which has sulfur in the side chain, which is cysteine and methionine. Okay, and if I go back to the structure again, as you can see here, this is the cysteine, but it doesn't have a thioether linkage, and there's methionine. So see, this is the thioether linkage because it has a CH2 here and a CH3 here. So one R group is CH2 another R group is or like CH2 like ethane, and other thio group is methane. Okay, so methyl group or ethyl group is there. So yeah, so the so the answer will be methionine. And since there is only two amino acids, cysteine was not here, so only option would have been methionine, even if you don't know the structure difference between cysteine and methionine. What is the sorry? What the amino acids used in ketogenesis to generate acetyl CoA during catabolism is arginine or glutamine, leucine or lysine, glycine or proline. This should be also this is, now this was not well taught in the class. I don't know why this question was there, but so to understand this, we have to understand two concepts, which is known as ketogenic amino acid and which is known as the ketogenic amino acid and the glucogenic amino acid. Now, ketogenic amino acids are the amino acids which form acetyl CoA during its catabolism, because amino acid catabolism was not taught in that class. So, but it is just to know this thing that ketogenic amino acids are lysine and leucine. There are glucogenic amino acids which are these, and there are amino acids which comes in between glucogenic as well as ketogenic amino acid. So these are this amino acid. Now, as you can see, ketogenesis, so when I say using ketogenesis to generate acetyl CoA will be the ketogenic amino acid. So the correct answer, of course, will be the lysine and leucine. Okay. And I have in next slide, I have actually kept one uh, like kept on a schematic that how amino acid catabolism work. If you are interested, you can go and have a look at this how the amino acid catabolism actually works. You can also go in Dellinger and see that how amino acid catabolism actually works. What is the pathway? Okay, or I can also show you now because we have enough time. Uh, so this is the one. Yeah. So let's go to the metabolic pathways. Uh, and then Let's see. The urea cycle. So this is a kind another part of the amino acid catabolism where you form the urea actually. It is known as urea cycle, but it's not scope up in this close to course to go detailed of all metabolism. So yeah. So again, yeah, this is the figure I was talking about. So this particular amino acid they can form like both ketogenic. So these are ketogenic and kind of in the boundaries. So as you can see that some of them can be common to both. So when the phenylalanine can both generate ketone body as well as phenylalanine can form fumarate to give rise to the in the glucogenic pathway also. So this was what I was only telling. So you can go to the 711 page of uh, the Leninger book and you can see the you can save details of this how this works. Okay, and there is a several pathways that how do you do. So I'm not going to in details of this. Okay, I just wanted to show it. Here you can see that ketogenic and glucogenic amino acid is there. Okay. 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 So the amino acids present in the interior core of the globular protein is each of them. Okay. So this is again a concept about this is again a very interesting concept, and so I'll just give me 1 second. I'll just put my laptop in right on the laptop. So again, this is a concept. This is a conceptual question. Now this you won't have to remember this kind of questions. Okay. So just think of a globular protein. So what is like present within the interior core of the globular proteins as well as the surface of the transmembrane region. Okay. Now think of in an integral membrane protein. Okay, in an integral membrane protein. Now think of an integral membrane protein. So this is my membrane. This is my cell membrane. Okay, this is my this cell membrane. Now the cell membrane, as you all know, that they are made out of this cell membrane. Our cell membrane is amphipathic, right? So it has this polar side chain. These are the polar, and these are the non-polar. Correct. Now you think that now we have something like this everywhere. Correct. So we have something like this everywhere, like this. And we have this polar chains outside here. So here also we have this polar. Now, if a protein wants to go inside, say if my protein, I make it in green. So if my protein wants to get in here, what it has to first do? It should not like, so this part hates water. So you can, as you can see, if you drop a drop of oil in water, it starts floating, correct? Or if you just put just even try once that put a drop take a drop sorry water some in some sorry take a some oil in some dropper, put it inside the bucket and just press there. You'll also see that it will form this small round balls like of this in the water, correct? So this happens because these are hydrophobic molecules. These non-polar things are hydrophobic molecules. So hydrophobic molecule doesn't wants to go inside, so doesn't want like water. So now this part is charge, some polar part, correct? Or if you think of the globular proteins, if you think of the globular proteins, outside it is very polar, but inside it has to be non-polar, correct? So if you think of globular protein, So what now inside? So there would be only the hydrophobic part in the part will grow inside. Correct. Because this part doesn't work. So this part should be hydrophobic, and to be hydrophobic, it has to be non-polar. Because in a globular protein outside was there is mainly water, right? So water means it is polar. Waters are H plus and OH minus ion. So it's outside it is polar. So of course the globular protein inside it has to be a hydrophobic environment inside. So the correct answer of course will be amino acids with non-polar hydrophobic side chain. Amino acids like say this amino acid, valine, isoleucine, leucine, which are very hydrophobic side chains. They don't like at all water. Those amino acids will go inside this part. And outside part, there will be those uh, amino acids that are more likely to be polar, like lysine, arginine, and those. There are very few cases where you will see a polar amino acid inside the core of the protein. Okay. Okay. So in thin layer chromatography technique, the phase returns the movement of the molecule during interaction of sample is solvent phase, stationary phase, mobile phase, or liquid phase. Now, what is thin layer chromatography? Now, so in thin layer chromatography, as you can see here, so if this is a TLC plate which is formed with some silica gel. Now, silica gel is a so it's formed with silica gel. Now, silica gel is a polar thing. Now, this kind of chromatography thin layer, always remember it based on this concept polar and non-polar amino acid. Okay, polar versus non-polar, correct? Now, what happens when I add this non-polar solvent? This is a solvent. The solvent is it's a non-polar solvent, right? This non-polar solvent, when it is when it condenses, just 1 second, I myself have got a doubt. Actually, I myself have got a small doubt. Yes, I'm just clearing. Yeah, CSI page 4 and the liquid mixture. Yeah, correct. I was correct. Yeah. So this silica gel is also like SiOH4, which is a polar thing. Okay, which is a polar thing, and polar. So now since it's a polar thing, so polar amino acids will try to get bound to the silica things, and as the non-polar solvent like chloroform, it goes up as part of the capillary action. The non-polar amino acids, the non-polar amino acids will tend to move with the solvent front. Okay. Now there would be like tension between, as it's discussed in the course also, there will be a tension between like forward and reverse. So the silica gel will have a force tend to attach to the silica gel. So this will have like tend to attach to the silica gel, and this will be the go to the solvent from. So the net force, the RF, will determine the net force RF will determine the presence of amino acid in the front, correct? So now what is so now the silica is stationary, right? It's a stationary phase. And solvent is the solvent phase, correct? The silica is the stationary phase. So now what is retarding the force? Of course, the silica is retarding the force because it's pulling the force down. So that means the in chromatography technique, the phase retards the moment of molecular interaction of sample is of course the stationary phase, correct? Okay, the amino acid iodinated hydroglobulin. Again, I don't think it's something I have to explain this. So uh, that thyroxine is also known as the diiodotyrosin. So as you can see, this first it will form a monoiodotyrosin. This is a tyrosine molecule. This one, and is this tyrosine? First one iodine is added, then two iodine is added. So this thyroxine metabolism T4 to T3 and reverse T3 is there. So this is again you can go and have a look on this particular part, that how iodine, like how thyroxine metabolism is done. It's again it's not possible to teach in this course. So then that will be T4, T3, triiodo uh, thionine, so 3,5,3,5 tetraiodothermin, the T4, and the T3, 3,5,3 triiodothermin. There is also above a reverse thyroidine, where we'll have like, as you can see, the in this thyroidine, the two iodine group is formed in this tyrosine, but as you can see here, it is from here. So yeah, so this is so basically the thyroglobulin protein as iodinated tyrosine, it will be tyrosine. Okay. The protein structure solely stabilized by covalent bonds would be, of course, the primary structure, because in primary structure, as I was shown you, will have this like the R group, R1 would be connected with this CON sorry NH group with another R2, correct? Small thing check here. Yeah. So this CONH will be connected R1 and R2, correct here. So then, so this is the peptide bond, and this is totally stabilized by this covalent bonding. There is no interactions between R1 and R2 in this case, correct? So the correct answer is primary structure. The major driving force of protein folding is ionic interactions, hydrophobic interaction, electrostatic interactions, salt bridge. Now, this is a kind of like uh, the factual that most of it is formed by hydrophobic interactions, because as you can think, as I explained in a few slides back, that all the proteins that have this kind of tendency to uh, like the formation of the structure is mostly the hydrophobic amino acids will be in the core, and the hydrophilic amino acids will be at the site, okay, of a globular protein. So hydrophobic interaction is the major driving force and during this protein folding. Okay. 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 The reagent used in the cleavage of the polypeptide chains after aromatic amino acids is chymotrypsin. because there was a list in the course that was taught that wherever the bonds have to be broken. The methods to use determine 3D structure of the sorbent protein. Chromatography, no, because chromatography we can just think of like what is polar, unpolar, or like based on the volume, correct? So it gives, so chromatography mainly gives us the either size and polarity. Okay. Mass spectroscopy. Mass spectroscopy is mainly used to determine, not sorry, not structure, but determine the sequence and modifications in the protein. So many, as you know, that many proteins have like uh, have tendency to phosphorylate, methylate, ubiquitinate. So these things you can also found within the mass spectroscopy. In the mass spectroscopy, yes, with this we can determine the structure in soluble phase. Correct? So the correct answer is NMR spectroscopy. Which factor does not modulate the binding of oxygen to hemoglobin to produce oxyhemoglobin in lungs is high pH. As we know, in high pH the binding changes, correct? The relatively low hydrogen plus, of course, again the low pH, high pH issue. The low 2,3-bisphosphoglyceride. Now 2,3-bisphosphoglyceride is not taught here, but it's a controlling factor again and increasing the temperature. And uh, correct answer was not modulate binding of oxygen to hemoglobin. I am just considering it. This. I'm just checking if correct answer is given because two, 3 responses. I think there is a problem this in this. So the correct answer is increasing the temperature, and yeah, fine. I just had a confusion about 2,3-bisphosphoglycerate. So actually 2,3-bisphosphoglycerate interacts with the interacts actually with, it's a anionic compound. So it's present in this human blood cells and it binds with greater affinity to deoxygenated hemoglobin. So hemoglobin generally uh, dislodges it and uh, basically, yeah. So it interacts with deoxygenated hemoglobin beta subunit and decreases the affinity for oxygen and allosterically promotes the release of remaining oxygen bound to the hemoglobin. So it's used as a allosteric effector of the hemoglobin. Okay. Okay. And yeah. So the GTP protein, which is involved in pinching of clustering code, now this was also not taught. So only it was taught that during pinocytosis, this clustering proteins, they coat the vesicles, and they now a protein known as dynamine use a GTP bound molecule. So this sorry, use GTP energy to constrict the constrict the membrane here, like as you can see, you can think of a uh, like how do I say? You can think of a thread. It's like it's a thread. It's thread going around the vesicle to more and more constricting, and at some point of time, as you can see, this will constrict and become like this. Membrane will fuse, and this membrane will become like this, and the vesicle will come out like this. Again, I hope this will be taught in the vesicular transport chapter in the next week. So the correct answer will be dynamic. Pathogen cells and unwanted cell debris are scavenged from the body through exocytosis. So exocytosis is throwing out things of the body. Endocytosis, the kind of this. Pinocytosis also is a kind of what I say, taking things inside, but it generally takes up from extracellular fluid, but not the phagocytosis. So phagocytosis is little different. So correct answer is phagocytosis. Pinocytosis is a process when like cells takes up fluids from from the environment. Okay, this is known as pinocytosis. And in phagocytosis, generally it's the cell is directed and take up large particles. Okay. The main difference between pinocytosis and phagocytosis is that phagocytosis large particles like bacteria, cell debris, whereas pinocytosis small particle size, like some molecule, some fluid or something like that. Okay. This is sorry, twice it has been pasted. During injury and wound, platelet release a mitogen known as, of course, it's kind of like written. So platelet derived growth factors. And also, it's a no-brainer, correct? Okay. So molecular technique used to identify antigen in biological fluids. Of course, the thing is ELISA. Southern blotting is to detect DNA from DNA. Northern blotting is used to detect RNA chromatography as you. There is also something known as Western blotting. You can also use Western blotting to detect antigen also, by the way. Western blotting to detect basically protein, because antigen is also most of the time protein to detect. If the antigen is a protein, detect protein. The purpose of blocking solution in ELISA is to stopping the enzyme reaction, to block all binding sites, to prevent the specific binding, to block the remaining protein binding sites on the wall. Okay. So blotting buffer is of course to block the remaining binding sites of the well. So what happens if you consider any kind of blocking solution? It's human. So if there are many binding sites here, change the color so that if we have many binding sites here, now there could be specific binding and there could be non-specific binding, correct? Now specific binding should have a very strong affinity. So what the blocking solution does is it block all the sites literally. Then we add the antibody. Since the antibody has a very high affinity, so it basically removes the blocking solution and gets the proper binding, correct? So that now all the other binding sites are blocked except the specific binding site. So only the antibodies will have the specific binding site. Other binding sites will be blocked by the blocking buffer. So block all the remaining protein binding sites on well. Okay. Just water. Which of the following is not correct regarding thin layer chromatography at TLC? Let's see. It's based on absorption mechanism. Yes, it's based on absorption mechanism. It depends on the functional group present on the light. Yes, it depends on the functional group. It's because polar versus non-polar, correct? This is what we want to see. The retention factor is unaffected by the temperature and thickness. No, this is not correct because retention factor 2 is affected by the temperature and the layer thickness. If the thick layer is too thick, then it will have more tendency to settle down than that, and temperature also will affect because temperature will increase the vaporization of the fluid, so this will also affect the. So this is our correct answer. Polar compounds will have low detention values. Yes, polar compound will move to the solvent from and then non-polar conditions after. So the correct answer is retention factor. This one. The site amino acid site chain most vulnerable to oxygen reactive is again a factual question. It's lysine because it has in each group there. The types of saturation curve so is hyperbolic, sinusoidal, sigmoidal, linear. As you can see, so this kind of binding is known as for hemoglobin to oxygen. This kind of binding is known as cooperative binding. So this kind of binding is known as cooperative binding. Okay, so cooperative binding means that as the more so the hemoglobin is alpha 2, as you know, the hemoglobin is alpha 2 beta 2. Hemoglobin is alpha 2 beta 2. Now you can think hemoglobin of like this. This is alpha alpha beta beta. And if one subunit, so cooperative binding means that if one first binding subunit KD, if it has a KD, so if binding of first hemoglobin or first ligand in this case, if the binding of first ligand has a dissociation constant of KD, then the binding of other constants, which like say this is KD1, then binding of other constants. So if I write it again, the binding of other constants one, two, 1, 2, 1, 2, 3. So lastly it will be this. So alpha, alpha, beta, beta, alpha, alpha, beta, beta, alpha, alpha, beta, beta. Okay. So if in this case there will be two bindings. In this case, three. These are yellow. I'm thinking about ligand, or in this case, hemoglobin, and this is kind of known as K. Fine. So if this is KD2, if this is KD3, if it's KD4, the KD1, so this kind of association, so this will be higher. So this will be like always less then KD2, less than KD3, less than KD4. This means that if one molecule of hemoglobin binds, this facilitates binding of the other molecules of hemoglobin. So as you can see here, so then at first the binding will be slow. Then as soon as more and more hemoglobin molecules binds, it is going to, then afterward, after at a point of time, it will get saturated because there is no no hemoglobin, no binding site. First, is this a substrate concentration? Right? No, what is so it's from so as you can see, it's a low affinity state, it's a high affinity. So this is transition state from low affinity to high affinity state. Okay. It's kind of known as cooperative binding. So it's a, of course, a sigmoid curve. The correct answer is sigmoidal. Yeah, so that was all the questions I had to discuss for this particular week. It was little factual, so I didn't had to spend much time for this week's. I couldn't. Ideally, I should spend 2 hours. So okay, if is there any doubt regarding regarding now? Anything you have to ask? So I can show you one. So this kind of thing with hemoglobin oxygen binding affinity. I can show you one image. Hemoglobin cooperative binding. Show you one image. Yeah, you can see here. Uh, that's fine. As you can see here, as soon as the more and more HbO2, HbO4, HbO, so as soon as more and more oxygen is binding, the binding affinity is increasing. Binding to the oxygen affinity is increasing. So oxygen binding affinity was low and it was high. So that's why the dissociation curve will be like this. Okay. As the oxygen partial pressure is increasing, the affinity is increasing, and at the point it's getting saturated. So it's a sigmoidal curve. Okay. In case of fetal hemoglobin, it's like a little sharp curve because the affinity is much more. Then there is my in my yeah, and myoglobin also, because myoglobin is a it's not capable of cooperative binding. Okay, it's made up of single peptide. That's why it's sigmoidal. Anything that is cooperative binding will be sigmoidal. It's not sigmoidal, it will be logarithmic binding. Okay. So yeah. Okay. So that was all I had to speak about today's session. So if there is no question, we'll end the session here. And thank you for listening.